I hope you're still okay for this last presentation. Well, in fact, this is a work which Patrick started a long time ago about habitat use in coastal Syngnathids of France. And the idea was to use citizen science to produce some individual-based observations. So, well, yes, I've been saying charismatic species broadly distributed in France. We've got about 11 species altogether. Well, exactly 11 species which have been listed. But we decided to add up to 9 more subspecies, morphotypes, and lineages that have questionable taxonomic status, and maybe some conservation and evolutionary significance. Well, as we've been mentioning earlier, so I'll go very quickly on that, ecology and conservation status is badly known, and non-professional scientists can provide a relevant approach, providing there is a standardized data methodology, collection from recreational divers. Anyway, the aims is to use this approach to enhance knowledge about habitat preference of Syngnathids in France. Okay. So, in fact, there's been quite a few citizen science projects or programs since a long time. First one started thanks to Patrick and his team and his association in 2006, and it's called Hypo Atlas. And the second one started in 2016, called Fishwatch Forum, and all used this individual-centered approach, each fish being related to its environment. So each photo is geolocalized, and the photos of the fish each specimen is taken. High resolution detection, maximum 100 meters of spatial resolution is required, and 17 environmental categories are used according to depth, substrate, biocover. And the photos are validated by volunteers, experts, so we're sure about the identification. So, as I said, we made a few hypotheses and taxonomic choices in order to detect whether structure and habitat preference vary according to biogeographical features and according to taxa, of course. So we had 4 biogeographical categories: marine or transitional waters like lagoons and estuaries, and Atlantic versus Mediterranean Sea. And 20 taxa were used for this study. Okay, so a few metrics were used, but I'll only speak about a few ones. And multiple correspondence analysis were made because it's quite difficult to see some variability. So here are the main results. What you can see on these photos is the variability of habitats where we can find the Syngnathids. And you can see that Patrick is always photographing everything he can find. So we've got a significant data set because altogether there were nearly 300 contributors and nearly 800 observations altogether, and well balanced between Mediterranean Sea and Atlantic coast, as you can see on this picture. Atlantic, Mediterranean. You can see the observations which have been made, number of contributors and number of observations here. And what you can see here is the number of counts which have been provided in Atlantic here. And these are departments, so it's more or less the geography across along the French coast. Of the Atlantic facade, and here you've got the Mediterranean story. So yeah, 4 biogeographical categories, 4 biogeographical lineages, 11 species, 9 morphotypes, and 20 taxa. So each photo is geolocalized, and the photos are validated by volunteers, experts. And the photos are validated by volunteers, experts. So yeah, the thing is well balanced, of course, but you can see that there is some sites where you've got many observations, and some sites where you've got many less, much less. So the kind of data that were collected is mainly from scuba diving, but also from shore walking and other methods. But most of the pictures come from scuba diving. And what you can see here is the depths. And in the Atlantic, most of the depths were between 5 and 10 meters, and a bit more shallow in the Mediterranean Sea. And green is lagoonal observations, and blue is marine observations. So the first story is that if you look at this graph, you can see where the observations come from. And if you look at this side, it's all the observations in lagoonal habitats. And here, this side is marine, of course. And these two colored graphs are only seahorses. We've got 2 species in France, and these are about pipefish. So what you can see very quickly on this graph is that for seahorses, you've got quite this similarity of the percentages of species, which is not the case in the Mediterranean. And what you can see here also is that on the Atlantic coast, the community structure is different, with some species being more abundant at sea than inside. And same story for pipefish structure, community structure. So there is an important disparity due to distribution area boundaries between Mediterranean and Atlantic, but also about habitat preference. So how can we work this out? Well, I've cut the presentation in 2 phases. The first one gives detailed descriptions, more or less species per species, and then, since it's very complicated, we try to do some multivariate analysis. So this one is, you've seen many graphs up to now, so this one is really a bad one just before eating. You must be difficult. Anyway, on this side, you've got the substrate preference, which is mud, sand, gravel, rocks, coralligenous genus habitats, natural debris, and artificial structure. So this is about habitat. And this is the biocover on this side. Of course, green is more or less seagrass, and red is algae. And here the blue is rocks, and the yellow is sand. And now on this column, you've got here you've got all the Atlantic habitats, and here you've got all the Mediterranean habitats. And here you've got the marine environment, lagoonal, marine, lagoonal. So wow, this is really complicated to disentangle all this, but and this is just for Hippocampus gutulatus, just the long-snouted seahorse. So what you can see from this very rapidly is that it's more or less the same on the Atlantic side. There's not a very big variability between habitat preference for this species, whether they're in marine or lagoonal environment. But you see, the story is not the same in the Mediterranean Sea here, where you see that there is a big variability of habitat preference between lagoonal and marine habitats, and there are also differences in biocover preferences. Also, you can see here the depths, and there is quite a bit of variability in depth preference too. So I won't go into details about this, but we've been doing this on hippocampus, And there again, you can see quite a lot of variability according to Atlantic, Mediterranean, lagoonal, and marine. So, not going much more into details, and there you've got the story about a narrow-snouted pipefish group, Syngnathids arcus group, and deep-snouted pipefish. And here again, the story is more or less the same. You can see quite a lot of variability. Once again, because it's before we're all hungry and tired, so I won't go into much details. And when I do, anyway, I get mixed up myself because it's too complicated. Okay. So what we've been doing is same story with same kind of work with worm pipefish group, and there again you see variability, which is really apparent from these graphs if you just look at the colors. So to summarize, we've been performing multiple correspondence analysis, and the first, these are just the two first dimensions, and the percentage of information which is extracted is low. We only found about 10% between the, collected by the two first axes of the multiple correspondence analysis. But what really came out is that in the Atlantic coast, you can see that stone and shell are opposed to fine sand habitats. And if you look at the main biocover, you can find red algae, which are, of course, more or less related to stone habitats. Bivalves here, living bivalves, and Zoostera covers. And here what you can see is that the Syngnathids and Hippocampus gutulatus are more in these areas, and while Lumbriciformis are in the stony areas, and Hippocampus and Hippocanthus of the Atlantic coast are more linked to shells and bivalve covers. And there's no clear depth preference between species, which was detected by here, because here, for example, 0.5 meters depth is here, 40 meters is here, 2 meters is here, so all this is mixed up, so there's not really strong preferences. And on the Mediterranean coast, well, we've got more or less the same distinction between rocks, fine sand, and sand, mud, or shells. And biocover is mainly Zoostera, algae, and Posidonia, and no cover at all. And here you can find the Hippocampus from marine areas, Mediterranean marine areas here, of course. And Hippocampus gutulatus is here linked to more hard substrates. And you can see that here there is more or less a depth gradient. So all this to say that the story is complicated, but what it means is that, one, citizen science has nevertheless proven efficiency. It's robust and sensitive to improve knowledge about ecology. This has been said earlier, but we've got quite a nice data set to confirm this. There's a strong variability of habitats among biomes, Mediterranean versus Atlantic, and biotopes, estuarine biotopes versus and lagoonal biotopes versus marine environments. And there is a discrete but significant control of environmental factors on the community structure of Syngnathids. But the limits is because we've got an unbalanced sampling effort, which is related to the citizen science approach. And also, the second limit is that we need to refine the statistical approach to better understand and take account of the small samples which are related to this kind of approach. Okay, nonetheless, this is probably the first study in France, and I think we've improved knowledge about this. And what I wanted to say is that if you want to conserve, and this is applied to marine protected areas, if you want to conserve properly Syngnathids, you have to have a big diversity of habitats. And don't think, well, I want this habitat only or this other. We need everything. But the proportion of everything needs to be better understood. Anyway, thanks for your attention. Thanks to all the non-professional scientists without whom the study wouldn't have been possible. Thank you.